Hi, I am Dr. Samira Kalaji and I'm working as a research associate in Professor Pier Giorgio Percipale's lab at NYU Abu Dhabi. Stem cells are like raw material for our body. They are able to transform into many different types of cells. In some cases, they can also fix damaged tissues. So researchers like us believe that stem cells may one day be used to treat serious metabolic and neurodegenerative disorders such as diabetes and Alzheimer's. Our lab is interested in finding the molecular mechanism by which stem cells undergo cell-fate decisions, especially adipogenesis, which is formation of fat cells, and neurogenesis, which is formation of neurons from stem cells. As a first step, we have to understand how stem cells regulate their identity and fate. Cell fate is accomplished by global changes, which are mainly achieved through a process known as epigenetic regulations. Our DNA is condensed into a structure complex called chromatin, and epigenetic processes control gene expression by altering chromatin structure. These epigenetic changes are happening in the form of chemical modifications or alterations in chromatin constituent or even in the 3D chromatin structure. We work with different types of stem cells, including primary mesenchymal stem cells that can make cells belonging to our skeletal tissue, such as bone or fat cells, and we also reprogram embryonic stem cells into neurons. We use advanced genomic techniques such as Hi-C, ATAC, and ChIP-seq combined with deep sequencing to study 3D chromatin folding during stem cell reprogramming. We use these techniques in combination with other molecular and cell biology tools such as microscopy to understand how cellular cytoskeleton proteins like actin and myosin regulate transcription and chromatin structure during stem cell differentiation into neurons and fat cells. Our next step to study genome changes in stem cells is to create organoids or miniature organs. Organoids contain thousands of cells that interact with each other in complex ways, making them excellent models for studying metabolic and neurodegenerative disorders. Currently, we are developing protocols that enable us to have fat and brain organoids in our lab. We believe organoid systems offer exceptional opportunities to improve and move the biomedical research field into a new level of personalized patient-targeted medicine. Characterizing the role of genome architecture has profound implications, not only for our research, but also our work has profound potentials for the practice of medicine, bioinnovation, and health in our society in UAE.